continue then on Saccharomyces cerevisiae for body yeast. I had given some introductory material the previous time on the size of the genome, various properties, the life cycle, budding, through the cell cycle, through the life cycle. You have different cell types. You have A alpha, and the A alpha diploid is the one that undergoes meiosis. Haploids cannot undergo meiosis, and that these properties of these cells are controlled by the mating type locus. And that is what I want to address now, how the mating type locus regulates these cell types. So another, of course, important model organism is a Schizosaccharomyces pombe or fission yeast. This divides by fission, right in the middle, and is very distantly related to Saccharomyces cerevisiae. So it's more closely related to yeast than it is to cerevisiae. It's a very interesting organism too. Okay, so what is the mating type locus? And I'll post these slides maybe sometime tomorrow. The mating type locus is the major regulator of the life cycle of fungi. So it's really a regulatory locus, controls many aspects of their life cycle, including cell types, but also sexual reproduction and pathogenicity in those fungi that are pathogenic. Here I'm going to concentrate on the mating type locus of cerevisiae, which has been studied for a long time and that we understand very well. But now we know that how the mating type locus works in many different fungi, and there are, not everything is like in cerevisiae. But this is like the prototype, this is the basics for understanding the workings of the mating type locus. Okay, so the topic of the lecture then is control of cell type specificity in Saccharomyces cerevisiae and combinatorial control. This refers to the ability of two different proteins to come together and generate or create a new type of regulatory protein, exemplified by proteins at the mating type locus in cerevisiae. Okay, so as I said, the mating type locus is a key determinant of the life cycle of fungi. Let me, of all fungi contain mating type loci, all of them. There used to be a group of fungi that people thought did not have mating type locus and a sexual cycle, but all fungi have the ability to undergo mating and undergo a reproductive sexual cycle. Ascomycete fungi, and I will tell you in a moment a little bit about them. Let me just get this. I need to get these images out here in my way because. So, ascomycete fungi have one mating type locus with two alleles. So, ascomycete fungi are fungi like cerevisiae or aspergillus that you have been familiar, but other more unusual ones. So these are morels that you see in the markets, are edible, or the truffles, the very super expensive fungi that form these structures underground, and this is the most expensive fungus there is, edible fungus. And then there are these that form these cup-shaped structures where the spores are produced. So those are ascomycete fungi or the sac fungi. And sac fungi, because they produce their meiotic products in these structures, these sacs. called asci or ascus. Okay, and the basidiomycete fungi have two loci, each has multiple alleles, though there are exceptions, okay? So basidiomycete fungi and ascomycete fungi are the two major groups of the higher fungi, and there are many others that are considered to be the lower fungi. Okay, and here are some basidiomycete fungi, typical mushrooms, but then there are other types of mushrooms like this, with all these fancy decorations, the star fungi, those that grow on tree trunks, etc. So it's a great variety. They are very colorful. These are the most diverse of the fungi. And these are the ones that most people are familiar with because you encounter them in forests, around trees, etc. So, because basidiomycete fungi have two loci and each locus has multiple alleles, remember an allele is an alternate state of loss. So that means that basidiomycete fungi have thousands of mating types, thousands. So the Mat locus in Cerevisiae controls cell type specificity and also in other fungi. In Cerevisiae, Mat locus has two alleles, and they are called Mat-A and Mat-alpha. Okay? And these alleles determine whether a cell is A or alpha or an A alpha diploid. So A and alpha, as I mentioned, are haploids, and this is a diploid. The mating type locus resides on chromosome 3. So the question is, how do these alleles determine the specificity, the cell type specificity? So here in this schematic, I show the Mat-alpha locus, the Mat-A locus, and in an A cell, you have both alleles, A and alpha. And the colors, they are shown in different colors to indicate that the information contained at alpha is different than that one at A, okay, as I will show you in a moment. So the three cell types have different properties. So A alpha and A alpha cells have different properties. So what are these properties? So before I tell you about the regulation of MAT, because it has to do with these particular properties of these cells. So alpha cells produce an alpha pheromone. Remember when I described the life cycle that I said that alpha cells produce alpha pheromone that is perceived by a receptor on the A cells, and A cells produce the A pheromone, which is perceived by the receptor on the alpha cells. And then when these receptors perceive this signal, the pheromone, then you form this structure called the shmoo. And that I described in the previous lecture. So alpha cells produce the alpha pheromone and a receptor for the A pheromone. So these are said to be alpha-specific genes, but they do not reside on MAT. These alpha-specific genes are dispersed throughout the genome. They reside on different chromosomes. That's what I said. The MAT locus resides at chromosome 3, but these alpha-specific genes are in any of the other 16 chromosomes in the yeast cell. Alpha cells do not undergo meiosis. And alpha cells express haploid-specific genes. So these genes, haploid-specific genes, I will tell later what they are. Again, they do not reside at MAT. They are located elsewhere in the genome. Okay, so just like alpha-specific genes are dispersed throughout the genome, haploid-specific genes are dispersed throughout the genome. So A cells they produce the A pheromone and except for the alpha pheromone. So the alpha pheromone and the receptor are said to be A-specific genes. They do not reside on Mat. These A-specific genes are required by these cells to make as A, and the alpha-specific genes are required by alpha cells to mate as alpha. Okay, A cells, like alpha cells, do not undergo meiosis. They express haploid-specific genes, and again, these haploid-specific genes reside somewhere in the genome, but not on Mat. Now, A alpha cells, they do not produce pheromones or pheromone receptors, so they cannot mate. They do not express haploid-specific genes. So A alpha cells do not express A or alpha-specific genes, and they do not express haploid-specific genes. They undergo meiosis. Okay? So in this, they differ from the haploids because they undergo meiosis, whereas A or alpha cells do not. Okay, so this is summarized here in this table. So you have expression of A-specific genes, alpha-specific genes, haploid-specific genes, whether cells undergo meiosis or not. So A cells express A-specific genes, but not alpha-specific genes, and they express haploid-specific genes, do not undergo meiosis. I want to emphasize that A cells, alpha cells, and A alpha cells all contain these genes. They have A-specific genes, they have alpha-specific genes, and they have haploid-specific genes. It's the same as saying, like in your liver cells, you have all the genes that are also present in brain cells, yet you do not express genes in liver cells that are required only in the brain. Likewise, in the brain, you don't require genes that are important for function of the liver. It's the same idea. Okay, alpha cells then do not express A-specific genes. They have them, but they do not express them. They express alpha-specific genes. They express haploid-specific